to the MVZ to sample the feathers from the collection for the work that they'll be talking about today. At the time they were living in Maryland, but they started a collaboration with folks from the San Diego Museum of Natural History who were doing resurveys of the San Jacinto Mountains as part of the Vernal resurvey project. Chris and Marilyn moved to UC Merced in 2013. Chris served as director of the Merced Vernal Pools and Grassland Reserve. I've learned that he just stepped down recently, so he's semi-retired. Still doing some Grinnell resurvey types of things in Yosemite National Park, actually. And Marilyn is professor at UC Merced. And the fun fact is that Chris Swarth is Harry Swarth's grandson. So Harry Swarth, here's field notes from Harry Swarth from 1908. And so Harry Swarth worked with Grinnell on these early resurveys, and it's really exciting that his grandson is there carrying on that legacy. And thanks to Christina, a little bit about Harry Swarth. So Harry left the Field Museum in 1908 when Grinnell invited him here to be assistant curator of birds. And he became the curator of birds in 1910. And then he left for 3 years to go to the LA Museum of History, Science, and Art. And then returned to MVZ in 1916, where he stayed until 1927. And apparently he and Grinnell had a little bit of a disagreement. There's some correspondence about why he left the museum and something about biological bird inspections for the biological survey. We're not really exactly sure what happened. I don't know if you know any of those details. But they had a little bit of a falling out. So in 1927, he went to work for California Academy of Sciences, where he stayed until his death. So anyway, it's great to have you guys here. We're really excited to hear what you've learned from our specimens.
Thank you very much. And thank you, Carla and Michael, for inviting us here to give the seminar and tell you our story about working on the centennial resurvey of the San Jacinto Mountains. So this is a tag team talk, and I'll go and give kind of a preamble and set the stage, and then turn it over to Marilyn. Okay.
No, you do it.
Oh, okay. So as Carla said, Joe Grinnell and Harry Swarth were colleagues for a number of years. Joe invited Harry in 1908 to come out to Berkeley, and 1908 was an important year. Obviously, that was the year the MVZ began, and as soon as Joe was selected as the director, he took off for 4 months in the mountains in the San Jacintos. This is the result of their work, the publication in 1913. But there's even more of a connection. These are their houses in Berkeley. This is on Benvenue. This is on Prince Street. So they both lived in the Elmwood. And they married sisters. So the Grinnells have figured importantly in the life of mostly my father and my uncle. But I do still keep up with some of the younger great-grandchildren. Joe Vernal had 4 children, and my grandfather had two kids. So the area of interest for this seminar are the San Jacinto Mountains, which some of you might not be familiar with. It's a relatively small mountain range, of course, in Southern California, circled here. Joe Vernal had a keen interest in Southern California. He worked on his PhD on the lower Colorado River, and then did quite a study in the San Bernardino Mountains, just to the north of the San Jacintos. But he was real interested in looking at the San Jacintos. Biogeography, geographical races, barriers to dispersal, these were all topics that really occupied his curiosity a lot at that time. The San Jacintos are located down here. They're the northernmost of what are called the peninsular ranges that extend down into Baja California. And then nearby are the transverse ranges, San Bernardinos, San Gabriels, and then the southern end of the Sierra Nevada. It's an unusual mountain range, not only because it's relatively small, but the elevational difference is huge. Palm Springs is about 450 feet in elevation, and the San Jacinto Peak is almost 11,000. If you've ever ridden the tram, you know how remarkable it is. It's just very steep on the east side. And then we have the Colorado Desert out to the east. So this is a map straight out of their UC publication, Zoology, showing the extent of the high mountains, desert to the east, dry country to the west, and then shown in color are 4 of the life zones, lower and upper Sonoran transition, and then a relatively small boreal section, which especially intrigued Joe Grinnell because there could be comparisons made between the avifauna of the San Jacinto high elevations with those of Mount Pinos, San Gabriel, San Bernardinos, and also the Sierra Nevada. A quick look at the field sites in 1908. They had 20 sites. It was a 4-month blitz. Work began in early May. There were 2 teams, Taylor, Richardson, and Camp to the north, and then Swarth and Grinnell to the south. And they traveled between these sites either by foot or by pack train. So theirs was a collecting expedition. So they were collecting birds, they were observing birds, they were also collecting a lot of mammals. But we can see here, this is the team to the south, and some of their effort resulted in 882 specimens by Vernal and Swarth, and you can see the dates of field work and the elevations and so forth. And then the team to the north was their effort. And some of these sites, they collected quite a few birds. I think about 1,500 altogether. And of course, these specimens are here in the museum, and they're what enabled Marilyn and I to do our work looking at changes in time over diet. These are western bluebirds. And of course, every specimen, I'm not telling you anything you don't know, has a great tag with the date, the location, and the name of the collector. There's your grandfather. And these ones were by my grandfather. The field notes were voluminous, and they're also a huge source of information. While the publication is quite detailed, the field notes hold a lot more. And this note is from Joe Grinnell, that's from Harry Swarth, but I underlined this note here. They found a raven as they were traveling on May 19th, and it was so unusual he made a note of it. Ravens are now all over the San Jacinto Mountains, as they are all over California. And Harry Swarth noted a duckhawk that he shot, and we took a look at this bird in the museum. That's a peregrine falcon. And then the next day he encountered, well, some of you can read about the most intense storm he'd ever experienced in the mountains, where it was so intense he wrote a couple of sentences about it. Photographs were real important as well. Field notes, maps, photos. This one of Toplitz Valley, high up. And then this brings us to Phil Unit and Lori Hargrove. I corresponded with Phil back in 2006. At the time he says, hey, we're mounting a resurvey of the Swarth and Grinnell trip. Would you like to participate? So I said, absolutely. Phil is probably one of the foremost authority on birds of Southern California and was quite familiar with the San Jacintos, but no work had been done there in 100 years. Lori received her PhD from UC Riverside under John Rotenberry, I think in 2010, and has been a key partner with Phil on the resurvey. So a little bit more on the resurvey, similar to the goals of the resurvey in Yosemite Park and currently in the desert, to retrace steps, estimate confidence in making population estimations, accounts, and tests for trends, and then to develop a new baseline. The San Jacinto work done by Phil and Lori is so detailed, as was the original work, that it forms a really solid base for doing more surveys on into the future. So you can see 3 visits over 6 years. The 4-month study in 1908 was brief, as it had to be, but now Lori and Phil have spent 7 years replicating that, so a lot more effort. And then some of the challenges. The survey methods mostly were done with a shotgun in 1908. Today we do point counts and different things. How much confidence do we have in the presence absence of species, working across the site and then also through time, and some of the other issues. Logistical problems that exist today, not then, was differences in land ownership. The San Jacintos has Indian tribes, various federal and state agencies, much more complicated to get access. So, now to the family part of it. We're sharing our personal history with you. when Phil said, yeah, come out and join us, I asked my kids, do they want to come? And they said yes. So this is my son here. In 1908, when we got to California, the first place we came was the museum, where Jim gave us a warm welcome and a tour. And so Evan and Nick were high school students at that time. And here's Evan looking kind of bummed out, but he's holding this peregrine falcon that his great-grandfather had collected in 1908. Here's the tag. So
And he no longer has such a ridiculous haircut. I thought he was going to California, he was going to look cool.
It's a little dated. So my son ended up bringing out 3 other high school friends and then a college friend. He kept going once he started college to continue to come out on his own, and I came with him several times. And then we talked, yeah, Marilyn, but here we are at a field site in Tockwitz Valley. This is the exact spot where Swarth and Vernal camped, different cabin. And Scott Tremore is the mammologist, Drew Stokes is the bat specialist, Gene is a botanist, and Brendan and Nick, the high school buddies. So here we are with Phil, my sons, and my daughter came out as well, my son and daughter, and they did a lot of mammal trapping. That was what they were tasked with, running the mammal traps. I'm a bird bander and a bird netter, so that was my role. And Phil employed some of the same methods that were used in 1908 to get to sites. Pack trains. They had volunteers with mules or burros and horses who got them up into the high country. But a technique that wasn't used in 1908, of course, was mist netting, so we did a lot of that. So I helped Phil run the nets and get the birds and ID them. And then just some of the very quick results, because I'm not here to tell you the in-depth results at all from this resurvey, but you can see some of the effort. They put out acoustical recorders to compare point counts versus acoustical recorders in terms of their efficiency and quality. Lots and lots of mist netting and field notes, and you can see the photographs. And here's Phil doing a point count there near Banning and a dusky flycatcher. And then if we look at a comparison between the effort on collections, you can see with birds, mammals, and reptiles, it's remarkable. We're concerned with their effort to study the birds, but at the same time, these 5 men were also intensively studying the mammals. They were both mammalogists and ornithologists, of course. The species collected, as well as the number of individuals. And, you know, a very comparable effort. Not that Phil and Lori were into matching numbers exactly, that wasn't the point, but they had a very specific goal in terms of specimens at each of the collecting sites. And then as with the Yosemite survey and Mount Lassen that MVZ has done, there was a lot of comparison of photographs and a lot of efforts to take the exact same photograph from the point where it was taken in 1908 to see some changes. This is Palm Canyon over near Palm Springs. And it's cool because you can see sometimes the same rock. Above Idlewild, which is the community in the mountains at about 6,000 feet. This is above Idlewild from Tockwitz Peak looking towards San Jacinto Peak. And you can note the greater extent of the montane forest today than then, and this is mostly due to fire suppression. And the expansion of the forest has meant for quite a few changes in the distribution of birds. Up at Tockwitz Meadow, or Tockwitz Valley, Lori Hargrove spent, I think, 2 summers trying to find this spot. I spent a couple of days, I gave up. She's very tenacious, and she did find the spot with the clue rock pointing up in the back. So in 1908, these are some of the birds that were collected, but these were the ones for which we sampled for isotope analysis. All resident birds, except for the gray vireo, which is a bird of great interest because its populations have declined markedly in Southern California. So we sampled them as well. But and so, like I said, then we made Marilyn get involved with this.
And they put her to work in the kitchen.
Yeah, well,
I'm only kidding.
so I'll let her talk about the isotope results. Here's your
Okay. Yeah, I have to say that. So again, this is a family project. So, you know, you have many projects as a scientist, and for all the women and the female scientists, I was looking at the campsite there where it was all men, and I'm thinking, so when did women start going into the field and going on these expeditions? And then, of course, you have children, so we've always taken our children on all the expeditions which I've had around the world. So this particular one was of interest to me because here's Chris's grandfather, who, of course, he never met because he died in 1939 or something, and this was disastrous for the family. And then to have the grandson and then the great-grandchildren actually participate in this. So anytime there is a study in our family, I think isotopes. And you have an isotope lab here and people who have studied that. So what we did was every time one of our young budding scientists went into the field, they made collections of plants, they made collections of insects. They went along with Phil Woods, Phil Unit, and bugged him to pluck a feather from mist nets, and also we spend time at the San Diego Museum and also here looking at things. So what I have to show you today is some very simple isotope data. What you see on this panel here are carbon isotope ratios of two sets of plants. The ones on the right over here were collected in the San Jacintos in 1908. And the ones on the left here were collected in 2008. So it turns out that these plants are not accessioned here, because you're a vertebrate zoology, but these are in the Smithsonian Institution. And right down, when I was in Washington, D.C., I just went right down to the museum and talked to Rusty Russell, who is the curator of botany at the museum, and he allowed the students to get access to the historic plants, and also his personal field area is in the San Jacinto Mountains, so this was very advantageous. So this particular collection, these are plants collected in a very similar area, because it's a very big area, and they're also very similar plants. So what you can see over here, carbon isotope ratios. I'm not going into detail on isotopes, but these are all what we would call C3 plants, a certain type of photosynthesis, both panels over here. And the average values for the ones collecting in 1908 are statistically different than the ones collected in 2008. And probably that's because the source of carbon dioxide in the atmosphere has changed. It's changed from when I was a girl, when it was about 320 parts per million, to now when it's about 400 parts per million of carbon dioxide in the atmosphere. The sample site that we're looking at, being to the east of Los Angeles, San Diego, has taken a big hit since automobiles came and the population grew in Southern California. So we knew going into this, and we expected to see something that was quite pristine from 1908, and a huge change that would happen over time. We also analyzed the nitrogen isotopic composition, and again, nitrogen isotopes on this, and the count of plants over here. So here are the historic plants, values of 2.7. The ones, again, the matching set, several per mil lighter than that. And this is, we were talking about your bees earlier on. These definitely are showing a shift in nitrogen isotopes over here. My idea here is this is because of automobile emissions and cows and everything bringing isotopically lighter nitrogen into the area. So this is now switching into modern plants and looking at nitrogen isotopes at the base of the food chain. So you go into an area like this and you'd say like, okay, pretty small area. I've worked continental scale in Australia, and I've been over the whole continent, and this seemed to me like, okay, it's a mountain range. And it was like, yeah, okay. So this is a comparison then of one collection of Fuller's Mill versus Santa Rosa Mountain, collected in the same year. And you can see here average values of minus 1.8 versus average values of 2. And it was like, is this significant? Well, it turns out that this is way high on the mountain, high up on a mountain. It's also in more eastern part of this study range. It's more arid in this zone over here. And Fuller's Mill is more on the western side of this range, getting probably more of the pollution coming in from the LA San Diego basin. So you look at this and you could say, okay, if I have historically matched samples, there's a difference in there. But if you go in there and you look at, there were how many sites? 17? 20 sites on here. It was like, oh boy, this was a challenge, because there's variation within them. This is a comparison now of the carbon isotopes. Here you can see Santa Rosa plants minus 27, Fuller's Mills minus 29. So just to let you know that the isotopic composition of CO2 that's in the atmosphere now is probably 1.5, maybe even 2 per mil, depending where you are, certainly in Berkeley. More negative, has a more negative value, less C13 in it now than it did 100 years ago. This is very well known. And when you're looking at this, is this a fact that you have different CO2 populations coming over, or there are other things when you collect a plant. So you're out there, you're collecting a plant, you're nipping a leaf, a needle, or whatever, and plants that are found in the shade have more negative isotopic compositions versus plants that are in the full sun have more positive ones because their stomates are shutting in the sun. The other thing that's happening is there's a wet-dry phenomenon. The wetter it is, a plant's able to open its stomates more. The drier it is, the stomates will close, and you have less isotope fractionation. So you're looking at this, and within plants that you have here, you have climatic signals of wet, dry versus shade, sun. You also have location on the range, and you also have where the source of CO2 is. Putting the data together for, this is from the collection now, combining both the carbon isotopes on the x-axis versus the nitrogen isotopes. You can see the individual data points here. The blue ones are 1908, and the red ones are 2008. And what you see over here is definitely this shift in nitrogen isotopic composition, but really no variation here for the carbon. But you know, when you start to think about this, this is definitely a complicated ecosystem. So the next thing we went to was, when I got involved, it was like we're collecting the rat out of everything, because you know, you send kids out and they bring back 20 samples. I like hundreds of samples. So what we have on here is nitrogen isotopes, and the rule of thumb for analyzing animals is you are what you eat. You have basically the same isotopic composition with just a little bit of a change in the isotope composition due to the metabolism of an organism. So for nitrogen, here are the values of the plants, here are the values of the insects that we collected here, and here are the values of the birds collected in one ecosystem. So in this case, you can look at this and say, if we measure the isotope ratio of a bird feather, you should be able to go backwards to the food that it's eating and backwards to the base of the food chain. Okay, so that's fairly basic stuff. And putting that together then on a carbon and nitrogen plot over here, this was the Tockwitz Valley ecosystem. So this was the one I went to. It's in the boreal zone. It was that beautiful meadow that Chris showed that picture over there. And you can see here are the plants that we collected. Here are the insects that we collected. The red symbols over here are the birds collected at that time. And the blue ones here are your museum specimens that we pulled off of here in 2009. So when you look at that, it was like, that's a pretty nice alignment. You are what you eat, a few per mil. And when you look at this, it was like, no matter, it was like you look at a compilation of data like that, they're not that different at all. But think about it, when you saw those photographs, Tockwitz Valley looked remarkably similar now than it did in the past. So this is not surprising. One of the things that we found here was we were able to get plans, historic plans. We came here, we got the historic birds, but what was missing were the insects over here. And I think that they're key. Of course you don't have insects here, no. And we couldn't find insects, and I don't believe anybody collected insects at all. So we made much more of an effort to collect as many insects as we could and look at their isotopic compositions. These are 4 of the different sites. Again, this was with our I have a breaking Joe Grinnell stick. No, our undergraduate scientists out here, and I put them in these ovals. So this encapsulates the standard deviation of the insects that you find in here. And what you can see over here is that you largely have overlapping isotopic populations of insects in these areas. Tockwitz and Round Valley, which is the black one, Tockwitz is the green one. These are the highest elevation, Santa Rosa Mountain. This was in the southern reach over here, and Kenworthy and Lake Hemet were down in the lower Sonoran part of the range over there. So when you start to think about what causes the variation in the isotopic composition of bird feathers, because that's what we're actually interested in looking at, I think you really need to not only nail down what's happening with the plants, but also what's happening with the insects. So we came here, we collected a number of species. Chris showed you the list of them. I don't always know that, when you think about things from the hindsight, it was like, how should we have collected these samples? In 2008 and 9, it was by Phil Unit and Laurie's design. They had permits for a certain number of birds. So we have actually a lot of the dark-eyed junco. We had 47 modern samples, 14 of the historic ones. The dark-eyed junco is omnivorous, consumes vegetation, consumes insects year-round. So, and we had a number of species where we had a pretty good component of modern ones and a pretty good component of the historic ones. So here's a nice jumbled graph. So we've got carbon isotopes here, nitrogen isotopes over here. The bluebirds are they're in blue. So they're let's see, western bluebird is this data point over here, and the historic ones are the 19 and the 20 ones are the modern ones. So here you're looking at we have lesser goldfinch, house finch, dark-eyed junco, western bluebird, and mountain chickadee. These are all common birds, which we have a lot of them over there. And what you can see over there is there's a lot of spread in comparing the two of them. When you go through and you look at this statistically, and you're realizing they're from this entire field area, and looking at that, the ones that stick out that are definitely different are the western bluebird, which you see in these darker blue values over here. The western bluebird, from the samples that Phil and Laurie studied, were ones that expanded their range and that are now found in more urbanized areas. And this is an example of a bird that definitely changed. It shifted downwards in its nitrogen isotope ratio and also expanded its carbon isotope ratio. I went through, and of all the specimens that we had, I assigned them to a trophic level based on known diets of these particular birds, analyzed things that were pure seed eaters, things that were mostly seeds, a little bit of insects, all the way up to, we're up here in the insectivores over here. And it was the insectivores where you definitely saw a difference between those species and their isotope ratios in 1908 versus in 2008. And in this particular graph over here, what I've done is I separated them from ones that were found in the east of the range versus the ones that were found in the west of the range. There's the p-value for comparing the nitrogen isotopes. And again, these are your historic birds over here. The rest of them, you didn't see a difference between where they were collected in the range, and I'm going to get onto the comparison of 1908 and the current ones. So this is the mountain chickadee. Here you can see the number of samples, 38 modern, 15 historic. Varies by season, eating seeds and insects during warm things. So we also took our data and we used SIAR modeling. It's stable isotope modeling, where you put your data into a computer program. This is Bayesian statistics, and here's 3 different types of potential food sources, insects and various plant material for the modern mountain chickadee, and you can see they're relatively eating about the same abundance. And here's the same data using the same program, showing that historically there was a greater proportion of seeds or plant material in their diet. And again, that's the sort of thing where you get really very small isotope differences, and when you get them, what people do is in general put them into these Bayesian statistical models. Now the models have all sorts of problems with them because we didn't collect insects in the past. And so what I had to do from there was to look at our modern insect data and our modern plant data and sort of estimate what the isotope ratio of the insects were. And the good thing about these models is you click a number and you can run the model very different ways. Another bird that we had quite a few specimens on, here's the western bluebird, 35 modern, 12 historic. And again, you can see it has this shifting of whether it's eating insects or seeds. We also, for the bluebirds, we measured a suite of sulfur isotopes. So sulfur isotopes gives you a third tracer, potentially, of what might be contributing to the environment, I would say the ecosystem. It's not necessarily a diet tracer in this type of a study. So the differences in sulfur cycling between 1908 versus 2008 would definitely be combustion of fossil fuels and a lot of SO2 released into the air from combustion. Here's the data for the historic bluebirds versus the modern ones, and here's also the nitrogen data that I've showed in different graphs over here. So definitely by looking at this, looking at locations, one of our conclusions could be that there are definitely niche changes, particularly with the western bluebird. So we also took the bluebird data and did some of the isotope models on there. So one of the findings from the San Diego Museum study was that western bluebirds expanded their ranges both up and down. So we compared bluebirds collected at greater than 2,000 meters versus ones at lower elevation. And you can see from this trending over here that, that there is probably, there's a hint, it's Bayesian statistic, that perhaps the ones at higher elevations ate more insects, but that's a hint. When you look at that and compare that with the modeled data that you see from the 100-year bluebirds that were collected at all elevations, you're seeing that's definitely a different trend. More insects now and fewer insects in the past. So one of the things that has happened in this area is that even though you have the biogeography and the range is the same, and this has been very well preserved, on the edges of this ecosystem, you have intense urbanization coming in from Palm Springs on one side, and you have the onslaught of the gases and everything that's happened from the LA San Diego basin coming to affect the chemistry on the other side. So another isotope plot over here. So what I did was I compared the carbon isotopes from the 2008 samples with the 1908 samples. And you can see them, the names are going along here. California towhee, dark-eyed junco, house finch, Cassin's finch, and lesser goldfinch, and calculated whether or not these were statistically different isotopic compositions. So for these are all herbivorous species, primarily herbivorous species. These two were statistically significant, the rest of them were not. For nitrogen, it's the same species over here. There were two of them that were different, the dark-eyed junco and the Cassin's finch. So you're looking at that and was like, modest change, Marilyn, you know, really is there anything into that? Then moving on to the secondary consumers, these are ones that definitely have a greater percentage of insects, known insects in their diet. And they're written in here by code over here. The one that shows a change in carbon is the western bluebird. And the other one is here, FS, what's that?
Fox
Sparrow.
Fox sparrow. Good thing we have someone who knows, okay? And it was like, is there any change here? It was like, not really. You know, you start on the pathway of a study and you hope you're going to see big changes. Not really for carbon. Now, when we get into it, here's the nitrogen differences over here. And I think these, when you're looking at it, here they go all the way along over here. So what you definitely see over there is that even though we know that CO2 in the atmosphere is very different now than it was in 1908, isotopically, concentration-wise, because this is such a complex ecosystem, you don't see that little difference that we saw in the plants. But for nitrogen, in those organisms that are relying on insects, you definitely are seeing a change in the nitrogen isotopes. So the question is, when you start to look at this, is this a change? When you see a shift in nitrogen isotopes, it can mean a number of things. One could mean that it's feeding at a different trophic level. It could mean that birds then had more insects in their diet than they are now. The other change that we talked about with the plant values is that if the whole isotopic baseline shifted downwards because of the contribution of pollution coming in, then that would shift the whole ecosystem down. So what I put on here is, let's see, open symbols down here are 2008 and 9 birds, and these, the solid ones are the 1908 birds over here. And what you can see over here is here are the same species collected in 2008. These are the mean values. These all of them, these are individual mean values of the different specimens that we analyzed. You can see right now, these two in a modern time almost sit right on top of one another, even though we know that they actually eat something different. Whereas in the past, over here, these read the isotope book, and they're about almost, not quite 3 per mil difference, but in general, this shows that these definitely were feeding at a higher trophic level. So when you come down to it, we are looking at combinations of all sorts of things. We're looking at, did the diet of the animals change from one time to another? This is what all, if you're into feeding ecology, that's a big thing that you want to know. Or were these environmental changes? And every time I've done a study in my life, it's never been one or the other. It's usually a combination of both of them. So there are ways we could take these samples, we could get to more complicated compound-specific amino acid analysis. I mean, I have hundreds of these samples. That's a possibility. The other possibility is to look at the environment where you know you've had changing fire history, urbanization, and of course climate change. So when I go back and I look at this, and it was like, oh boy, and I have reams of data, thousands of data points here, and I'm sure some of you have had thousands of data points that you had to look over, and you realize this is a very small mountain area, but the complexity over here of having this east slope over here, right next to Palm Springs, you have a big rain shadow coming over here. You have all your life zones, your elevation zones. We looked at all of the isotope data as it relates to elevation. We found no relationship between elevation or altitude and isotopes in mammals, birds, or plants. And so it was like slightly disappointing. But there is an east-west thing that's, I think, is important. So when I went back and I used this was PRISM data, right? So this is modeled data over here of precipitation, and sorry it's in inches instead of meters, but we're Californians, so we know inches. This is the 10 years preceding the samples collected, 1908, and this is the 10 years preceding the 2008 samples for the western part of the San Jacintos versus the eastern. So now that I live in California, I know all about El Nino years. So there are years when it doesn't rain hardly at all, and there are years when it rains big time, okay? We're in this time here. So you look at this, in 1908, there were very low rainfall years followed by 3 or 4 high rainfall years. You can see variations in the recent times. Here's 1998. That followed. That was an end of a big El Nino year as well. And so when I go back and I look at this historic data, I am not looking at a general trend of aridity or climate as you go through time. Because I have 2 points here. I'm looking at an El Nino signal possibly going up and down, and that's a level of complication that you have in California that you don't have in other places. It could also be a level of beauty. When you think about it, what is the effect of El Ninos over time, and could you find it? We do not have that amount of data to say this, but oops, this has done some funny things. Look at that. Bill, you're killing me here. I'm going to go on to this one over here since we're nearly out of time. So we get to the end of this. I see isotope changes. I've been able to come to your museum with Chris, because I was participating in the Olympics at that time, the Senior Olympics. I threw the shot put then. And you get samples like this, and this is such a valuable thing to study, but it's also complex. These are some of the causes from Phil and Laurie's report. Upslope changes responding to climate change, urbanization, disturbance, agriculture, fire, rebounding from past hunting, new avian colonists from the north, not like San Francisco people moving to LA. Downloop spread, more montane forest, the whole fire regime changing, increased density of all of these factors over here. And there was a whole page over there of factors that were also negative factors. And when you come down to it, this is a picture there, and it was like, this is my daughter when she cut all her hair off at this time. I was like, I have children with weird haircuts. And there's my son, and here's Phil Unit, and here's Scott Tremore, the mammologist. It's interesting. It was like they just got right into the guts of these, because they take after their father, not their mother over there. And when I know I'm talking to museum people, but my goodness, museum conservation is such an important part of documenting this. When I talk to other people around the world, and they're going, you killed the birds? And it was like, you gotta get over that. These are such a valuable resource for everyone. I'm, telling the over here, preserved animals, specimens, an animal's diet, where it lived, its position in a complex food web, the climate and an environment. And I would like to personally thank everybody and Carla for allowing us to sample these important specimens and to hopefully make a larger contribution to this. We educated a number of, I had high school students, they went from collecting samples to analyzing them in the mass spectrometer. They ran my mass spectrometers, and they were actually pretty good at it. They learned about looking at data, making graphs. They went off to college. I don't know that they're all biologists, but anyway, so this was a great way in which to spread this kind of thing to a wider audience. And
any of the bird species have here migrate?
they were mostly resident. They were residents.
Right. Okay,
So the ones that we got from, so if we were in the field, meaning Chris or one of the high school kids, they went with Phil, and when he was misnetting, they took whatever he got. But when we went to the museums and the specimens we collected here were resident birds.
And sorry, it's a two-part question actually, just really quick. Then are you able to play the same game with feathers that you could with mammal hair, where you could sample sequentially and look at changes in diet
I mean, in different parts of the
in high
bird.
resolution? Yeah. I don't know
Okay.
enough about feather growth patterns to know whether that would be.
So, number one, we're allowed to sample body feathers, right? You want to sample a primary from a museum, that's a much tougher thing. So I work with Gary Graves at the Smithsonian. He will not let me touch those. He has to collect special specimens, and we measured hydrogen isotopes in 25 feathers and black-throated blue warblers. Oh boy, read it and weep, there's some variation there, but not with these body feathers.
Sort of a follow-up on that, are the data such that you could separate periods when they're territorial versus non-territorial? Because when they're territorial, they're going to be in one spot, so you would expect lower variation than.
Yeah.
Well, they're resident birds, so in general, they're in the same area year-round. Is that?
Well,
they're not migrating. There might
No,
be
it's
some-
not migration,
But
but
moving
if you have a
around.
territory,
Yeah.
during
Yeah.
the breeding season, you're going to be
Yeah.
in a very confined area, whereas
Right.
outside the breeding season, you're going
You
to
could
wander
be-
Yes,
around
that's right.
more. And if you looked at the variance
Yeah,
during periods when
Yes.
they were nesting
Mhm.
versus, post
Right.
fledging.
yeah. We know the date of when they were collected. But because we're analyzing feather, which is discreetly grown, you really get the time of molt, which
Right.
is often like just after the breeding period.
So it would just carry over.
Yes,
That's kind of
so
short.
if you were looking at a continuous tissue, you would see that.
So I think like NASA now and planetary science people have these maps of N2O concentrations, based on satellite data. So I wonder if you could incorporate, or if you thought about maybe, like you can see these maps where near cities there's really high concentration in it too, and then it sort of trails off.
Yeah.
But kind of like a heat map over the whole world. So I wonder if those data could be used to tease apart
To look at various
the
things.
pollution versus trophic shifts.
So, we really wanted to look at not just two endpoints of 08, 19, and 2000, and we wanted to look at points in between, because what happened is you can see cars coming up as I look at this data, and then you can see catalytic converters coming in
Yeah.
and pollution controls, and you see a downshift because the LA Air Quality Board does publish historic data, not in map form, but in, you know, tabular form over there. Things are going down over time, but it would be interesting to see that plume, you know.
For one of the species, I forget if it was chickadees, you showed a shift or inferred a shift in diet, and I forget if they were eating more seeds or less seeds. My question was, does that correspond with known vegetative changes in those areas?
Does that correspond to known vegetative changes? Let's see.
I mean, are there more?
So the vegetative changes would be definitely in the edges of the range, and also the fact that there are more forests over there. There are coniferous forests over there, and so that's not part of their. They're not eating.
Well, they're forest birds, but down below where the chaparral has been burned more recently,
Yeah.
there has been a shift away from chaparral to more open grass. But the chickadees are forest birds.
Right.
So the only change I would know of is the forest is now denser and it's expanded from what it was in.
So were they
100
eating
years
more
ago.
seeds or less seeds? What was
I think they're eating more.
I think, yeah, it's like, I hate to do this to you, but, let me just look real
and-
quick.
So I was just wondering if the
If they,
kinds of seeds they're eating are more
Yeah,
abundant.
I don't know.
Of course, the isotopes don't give you historic more seeds now. Okay, so this could be, we were talking earlier with the
fewer
bee
seeds.
legs, because you do have a shift of native to non-native vegetation that could be happening there, and you could also have a shift to plants that they're not eating versus plants that they were very well adapted to. I don't have that information. There was plants collected, but I don't know that there is an analysis.
I don't-
I collected them for
I got it.
isotopes, not distribution. John?
All of the birds got lighter in nitrogen 15, except the goldfinch. Is there anything special about the goldfinch that accounts for it deciding to concentrate nitrogen 15 in the 21st century?
What would you guess?
Carla?
Because goldfinches are seed eaters, and pretty much only seed eaters. So, you know, why that would be, I don't know, because all the other seed eaters did not do this. Do they ever not, I mean, they only eat seeds, don't they?
Well, no, they eat insects when the young are in the nest.
Oh, when the young are in the nest.
But that's a relatively brief period. And it's
yeah.
before they would be, right before they're molting.
Before they molt.
Yeah, before
Yeah.
they're molting. So whether or not their life, this is like I'm teaching life history and ecology, whether or not their life history changed slightly from one time, or where they would shift their diet, this is a question, and I don't know the answer.
But it suggests it really be worth looking at that molting,
Yeah, or
Period,
that molting
rather
period.
Right.
the broader
Yeah,
thing,
right.
because that's
Yeah.
when it's all
Yeah,
happening with the
yeah.
isotopes.
Yep.
Yeah, like I say, I know that from when there are the birds that, I now have a permit to salvage birds. If I looked at the isotope ratios of primary feathers, I always show my primary feathers, and secondaries and the rectrices over here, you really see, you compare the left and the right wing, they're identical mirror images of each other, which it should be, because they're coming out at exactly that same time. And for museum specimens, you're always doing the body feathers, because people will sacrifice a body feather. But if body feather doesn't turn over the same way as the other feathers, which the molt is very controlled, so they're not the ideal to look at that sort of measured changes. But a body feather that I have found is that it's actually that average signal that you're looking for. It's not that exact signal that you might want, but it is that average signal. So when I've looked over, again, black-throated blue warblers, Gary shot hundreds of them, maybe even thousands. And we analyzed all body feathers, but when we went into the full bird, that's when you could really see the changes that had happened in their diet over the time they were molting, or if they were moving here, slightly moving there. That's, yeah, but in a museum, you're not usually giving those out, are you?
Flight feathers? No.
No.
We get a lot of
But
requests actually
But
for
again,
that.
if you developed a specific hypothesis
Yes.
that
Right.
was geared to that,
Yeah.
and said,
Yeah.
could we get partial parts of one of,
Yes.
I mean, we do, we have a lot of discussion about this, actually, because we do get quite a few requests for
Yeah, see,
flight
you're shaking
feathers.
your head. And so when we had the, and again, salvage birds, I looked at sooty shearwaters found on the coast here. Oh my God, the shearwaters, they're flying from New Zealand, Japan, Hawaii, California, South America, and you measure those flight feathers, I have a signal of the entire Pacific Ocean food web.
And they
in
molt
those.
as they're
And-
flying.
And they're flying.
And it's a slow molt. It's
Those I have a standard reason, and I can find them. But you know, but those historically would be really interesting, because you are looking at, you know, a very long distance migrant feeding on various patches of the Pacific Ocean. For a resident bird in a location, you know, it's small, it's subtle, it's something that I get excited about, but none of you would. I get buried in these details where I probably shouldn't. So, okay?
Anything else? All right, thank you very much.